First and foremost, thank you very much for your participation. This space is a space created by the Aapresid Pest Management Network. This is focused on drama to action, as the title says, which were the lessons learned during the last season to put them into practice for the next season. And for that, we learned Ines Catalano, CONICET researcher and teacher, an agronomist engineer who is a researcher of agricultural zoology, Obispo Colombres. So without further ado, I leave you to them. Augusto, the floor is yours. Good afternoon. How are you all? We're here to talk about the leafhopper, and I think that the focus of this talk is very important because we all know what happened during the last season, the problems we had, and what is important, regardless of all the losses, I think we should take away some lessons learned to deal with them in the best possible way. There are many areas with the presence of the corn leafhopper, and it's important to change the way we grow corn, to plant differently the crop so that it's that relevant corn of our productive system, especially in the northern areas of our country. This is an element that we cannot take it away from the system. Basically, I'm going to focus on some aspects. You must have seen this pyramid a million times, but this is what happened last season as to the dynamic, the pest management, and how we should handle this problem. And here you can see the pyramid, where the basis of the pyramid is supported by the management of the vector population. We are not talking about a pest that is getting that is damaging directly, but it's a very limiting type of diseases. There are vectors involved, so we have to focus, especially these days. We do not have corn as a crop. We have to focus and manage those initial populations. I will basically present some examples of tryouts that we conducted in the north of the country practically a year ago. These studies spring from associations with different organizations that were not only devoted to strategies, but had a very strong foundation that was to evaluate the dynamic of this vector.
Cultivo.
And we have crop monitoring, weed monitoring, the study of the percentage of infectiousness, which is a key element when it comes to a vector. Here is an example of two situations that could be repeated in many areas where there is corn crops. This area has corn only during the summer. And so what can we find before this? We could have some winter crops. We could have some late corn, but the idea is to have corn concentrated in the summer. What was the situation of this pest when we started doing the studies? You can see in these curves, when we started in September 2023, we can see that in this area we had 23 leafhoppers per plant. And you can see that the dynamic didn't show evolutions until in December we found corn as a crop. And in January you can see how it evolves, and that needs corn to multiply and reproduce. It's a key product. And throughout the crop season for corn, you can see how the population of Dalbillus maidis, we've had approximately 1,500 leafhoppers per trap. So that amount of leafhoppers can be very important. Yes, no, but we have to take into account a very important issue, which is the level of infectiousness of those leafhoppers. And that study was done based on some studies which basically in that area we had 3% of the population that had presence
infectada, digamos, por espiroplasma. Y pueden ver que con el avance, digamos, del tiempo.
But over time, this leafhopper would feed on corn. There were many sick plants, and that situation evolved with more than 50% of the population that was infected, and this marks the dangerousness of the problem. And what happened was that it was very cold. There were frost cases. You know that in that area we have 2,000 leafhoppers per trap. So how did we determine a significant reduction of this producer that produces in cold temperatures? In September, we had 20 leafhoppers, 30, and now we have 63 per plant. Yes, but we've had a very important element which contributed to the reduction. If not, we would still be at 1,000 leafhoppers. And we are seeing this in the crops, and we're seeing how it has evolved on weeds and the reduction of the population. Here you see the freezes that have occurred in that area, more than 10. And one important aspect is that in July, we had low temperatures for a long period of time, which contributed to solving this problem. But we've had to incorporate a different strategies because it's helped, but it hasn't solved the problem completely. We have other areas, just to give as reference, and it's an area where corn is grown in the spring, and then in the summer it is harvested. And we see the predisposition for the increase of this pest. We can see in this area how we started with the same level of 20 or 25 leafhoppers per trap, and what you can see is that the fact that this pest has high availability of the crops has made the evolution more significant. In this area, when we grow corn during the summer, we have 200 leafhoppers, much more than in other areas. And what is my point with all of this? Well, that the crop systems that we have in each area have a very clear influence in the behavior of this vector and the dynamic of this pest. This area had as much as 2,500 leafhoppers per trap in April, and that's very high. And we can wonder whether they're dangerous or infectious or not. Well, here you can see the infectiousness of these insects. 70% in May. 73% of the population was infectious. In that area, if we had corn, corn was sick. So all those leafhoppers that are feeding off off that maize had the vector. So that's the situation. In that area, the frost was had a strong influence. When we started having this intense cold temperature, we see how the population dropped. But like I said, the cold temperature is an important element, and it did help us, but it did not solve all the problems, and it will not eradicate the problem altogether. And that's a little bit what we're seeing in the crops that we have in that area. Winter crops like rye and some presence of Dalbilus maíz, but with the cold temperature, they reduced their population. But we had one frost after the other in June for a long period of time, with temperatures below zero and intense frost. So here we have two areas that are quite distinct in the association of the crop. If you weren't here yesterday we launched the first data of a national monitoring network for Dalbolus mites, which are in the yellow traps on the map. The first data are for the second 15 days of June. For that period of time, 146 sites or towns that were analyzed, and with the participation of CREA, of INTA, of Aapresid. So it's an interdisciplinary group that has proven very important, and where we've tried to see the snapshot of what the pest looks like in different places, in different agroecological areas where corn is produced. So this is the summary of that monitoring network. And I wanted to emphasize the importance of this zone. In the north of the country, with the presence of the pest, and the categories, you can see them on the screen, the scale. You can see that the central area has less amount of leafhoppers, and in the south, very low levels. So what I want to convey with all of this is that we have a lot of leafhopper with a lot of presence. So let's start planning and let's start learning from one another so that it doesn't take a toll like it did on this crop season. We're still not planting corn. We're focusing on this. Wild corn is here. We've had the cold factor, which allowed us to control this wild corn. But you can see the amount of grain that we see on the floor, on the soil, and that's a ticking bomb. And that will be born and will be used as a reservoir or to multiply the pest. And another minor element is that that type of corn, that volunteer corn, is a source for all the pathogen complex of leafhopper. So we'll not just allow it to multiply, but also use to increase its level of infectiousness. It's not the same to have 100 leafhoppers with 3% of infectiousness than to have 100 leafhoppers with 60% of infectiousness. Obviously, the scenario is completely different. So we have to work together so that areas that are close to start the planting, they have to manage this issue of volunteer corn. It's not the same as, I don't know, with soybean, I can wait or monitor. With this, we see a pest and we need to design management strategies all the year around. So it's not just for the crop. It's from before and after the harvest. The monitoring will be a key element. We have to learn. This is not an easy pest to monitor because of how it moves, it flies. We have certain times of the day in which they will not be found. We can underestimate them. So it's key to go back on the field, to be there often, and to be there for this monitoring. I believe that this requires a greater professionalization of the activity. It's not just, going on my pickup and seeing whether I see it or not, or whether I find it. There has to be a monitoring of the adult leafhopper, of the female leafhopper. And so in this way, these key elements have to teach us and incorporate them and plan it for this crop season. We participated in the meetings of a model company there in Tucumán. And what they did was show how they plan for corn and how they will do the step-by-step to be able to face this problem. We are talking about an area where the corn will be planted in December, but there was already a previous planning. This is absolutely key. The other aspect that I wanted to tackle was the chemical control. Chemical control is, in a way, the tip of the iceberg because it does contribute, but what happens is that I have to start at the base of the pyramid. If I haven't started implementing strategies at the base of the pyramid and I just want to do chemical control, what will happen is what happened to us in many areas, where we tried to control this when the population levels were very high, so it was already out of control. So I have to first tackle the base of the pyramid and move on from there. Chemical control is important when we have a relatively manageable population. So what do we have for chemical control? We can treat the seeds. This has been established. There's a lot of experience in Brazil for treating the seeds. But this is a pest that covers the entire cycle of the crop. So the seeds, treating the seeds will only protect the crop in the first stage. What is the most sensitive stage for the leafhopper? So that will be a first measure that I can take to protect my crop. And what will happen with the seed treatment? The doses that are registered in Brazil are much more significant than the ones that we are used to applying. So there is a huge effort being made there by seed companies to make the necessary dose adjustment to start the crop season with materials that have a certain initial protection based on that technology. An example are some tests and trials that we did here with doses that are registered here and in Brazil to assess the performance of that tool. But it's that small window for that tool. We cannot expect to escape just based on that. And the monitoring, someone said that maybe 2 covers the whole treatment. No, I have to start seeing it and analyzing it to detect what the tool does. When treating the seeds, I can say that, okay, the corn I have has been treated like this, so I can increase the doses, but be very careful with that. If you make that decision, it has to be your responsibility. And there are many recommendations made by the companies. Why? Because in many cases, these practices can affect the physiological quality of the seeds. And there's a study being developed, which is a fantastic study that analyzes the effect of re-curing the seeds and what the effect is on the physiological characteristics. And you can see this for different doses, different characteristics. When we go there and evaluate and see what is happening, and we see hybrid and hybrid, we see all the different hybrids, we realize how the physiological ability has been affected. And we can see this up to 17 days after the treatment. When we have more time, when we do more time, we see that more hybrids are affected, there are more treatments that appear, and so this becomes a measure where we cannot assure that we will have a correct implantation when using it. So really, you need to be very careful with that part. As for seed treatment, we need to see what application can be done on the leaves, because once again, and what was mentioned at the beginning, I have to work with low populations. The last crop season showed this, and also in commercial lots. When the population was very, very high, there's a whole list of treatments, but we would go back and we had between 7 and 8 leafhoppers per plant. And using some insecticides, the control 3 days after the application you can see that all treatments resulted in more leafhoppers or same amount as there was initially. So in that situation of high pressure, the foliar application has to be handled with a different set of criteria. A situation with a more moderate incidence where we had 1 to 2 adults, I don't know if that's moderate really. But there you can see the different treatments, and we can see more efficacy and control. And by this we mean controlling 30 or 40 in the best case scenario, or 70% maybe. But we can see how when we start with a lower population, I wouldn't say low, but just lower, the essence of chemical control changes. Of course, there are tools that do show more solidity than others, but this is overall the situation. And I wanted to show this because we learned a lot from this campaign, from this crop season. I could say, no, I don't have any more Dalbilus. Maybe I have 8 or 9. I don't know how many people I told this to. Oh, no, the problem is part of the past. You have no more Dalbilus. And then you end up realizing that it wasn't over at all. So these were some tests that we did on the phenology of the crop and up to what point we should care for it or not. There's a witness there, but treatment 5, we did foliar application. And supposedly until V8, we were on the other side. But these were the results. Witness, treatment 2, 3, 4, and 5. And why did this situation happen? We had a high incidence where in February, which is when we reached V8, we had more than 500 leafhoppers per trap and 40% of the infectious population. And here you can see the performance. So what this has taught us is that the phenology will be highly associated with the population that I have as a baseline. That treatment that we've controlled until V8, we can see that there's a very clear decline in performance and in the yield and in the quality of the seeds. So that's a little bit what I wanted to share with all of you. Here you have the entire work team. Every time they close their eyes, they see a leafhopper because they've worked so hard and dedicated so much time to this problem. Thank you.
Bueno, buenas tardes.
Good afternoon. An additional speaker presenting a talk about the leafhopper. They are to blame because they invited me. Over 20 years, I have been working with leafhoppers for over 20 years. And among the leafhoppers I have worked with is the Dalbilus mitis. Some years ago, with the Laboratory of Agronomic Interest, I work, and my idea is what we did at the laboratory throughout all these years to see if we can give some small solution in this chaos the leafhopper brought about. Particularly, in the case of Dalbilus mitis, we have 2 working lines. One with gene silencing, and the other one is the search for resistant genotypes. So I'm going to divide my talk into these 2 sections. So the first one, the gene silencing. Some 8 to 10 years ago, we posed 2 questions. The first was that we knew of the high reproductive rate of this leafhopper that was completely evident in this last growing season. And on the other hand, we began to see the RNA interference technique that had a potential to be used in the farming sector to control pests, because it's species specific. That is to say, we can focus exclusively to a single species, to Dalbergus in this case. It's not going to kill the bees or controllers, just it's species specific. Besides, it has a low environmental impact. Very simply put, what is this technique about? I'm sure you have heard about it with the COVID vaccine. There were two types of vaccine that had this technology. Many freaked out with this, but it's very, very safe. So what we do is we invite into our organism double-chain RNA. Once this double-chain RNA gets into the cell deploys a machinery against this external RNA because it's already incorporated in the cell to defend against viruses, for example. So this machinery cuts off this double-chain RNA into bits. But on the other hand, the nucleus of the cell has the RNA messenger that will say, produce this protein. These small pieces of chain connects to the RNA messenger, that is a single chain RNA, and then it starts getting into smaller parts. So the RNA messenger, the message cannot be translated or converted into a protein. If it cannot be converted into a protein, and that protein is essential for a biological process of the organism, that function does not get fulfilled. That's the RNA technique. So under these two premises, how do we work at the lab? We raised the Dalbergus at a control situation. We took a pool of those Dalbergus mites throughout their life cycle from the eggs until they are grown, and we sequenced them. So we did a transcriptome. So in all the genes that are being expressed at this moment, that's what we are sequencing. So all this sequencing data was received, a bulk of numbers and letters, huge. I'm sure there are a lot of agronomist engineers here. So imagine you print all the Excels you've got, and a wind comes and all the pieces of papers fly away. Well, and now we have to put them all in order. So we do a cleanup, we do an ensemble, we do metrics to see everything is combined correctly, and this become a doctoral thesis of the Toro Palacios in 2018, a master thesis. As Victoria was doing that, Lucia, who was doing her doctoral thesis, set certain things clear. One was artificial feeding, that is to say, to get the Dalbergus mites to eat from a specific device with a preparation based on sugar, etc. So it fed very well. We'd put coloring, and so the digestive system was in color, and we could see they were living and prospering. And on the other hand, she studied into detail all the embryo development, the embryonic development. With that, she finished her degree thesis, and then she continued with her doctoral thesis. So we started putting all things together. Victoria's study plus her data. So the first thing that she did was to look within that transcriptome the gene related to the RNAi machinery, because if we did not have all those interferences, there was not even a starting point for experiments. So we saw the Argonauts, the RSD2, etc. And then the interest gene that was bicaudal C, because we knew this from other species that acted on the fertility. So we characterized it, we knew where it was, where the gene was. We put up we ensembled our double-chain RNA, we injected it to the female leafhoppers. We left those female leafhoppers to recover, because although we used very thin needles, we had to put an injection into the abdomen of the doublets, and we let them recover. We put each female with male, and in each of these cages there is a couple, and every 48 hours we change those little cages. We change from cages, and we count the number of eggs. Besides, that plant gets preserved, because afterwards we see if they get developed and they become nymphs. This worked out. We silenced it. We could see the stats later on. But we cannot be injecting leafhoppers as we were injected the COVID vaccine. That's impossible. We needed to be able to silence by feeding. So we did all the same, but giving the double-chain RNA through the food. And we added the double-chain RNA. So we did the same procedure. We numbered the ovipositions and eureka. When this worked out, this was a big success. This that I'm telling you in just 5 minutes meant a lot of testings until it finally worked out. In this first graph, you see the number of ovipositions of a control trial is in black. Then we have the females, the silenced females, with 2 different doses, but practically, silenced by injection as it is in this first graph, we have no number of ovipositions. The second graph shows that it's statistically significant. The diminishment is significant, but not as much as in the previous case. Below you see some embryo. This is a control injection and feeding. And you can see that the embryo, if there is an embryo, if there are ovipositions in the injected females, they did not develop. The embryo did not develop. So this is some of the things we like doing it at the lab. But in order to finish and prove that what we were affecting was what we wanted to, and that we can be certain that the gene acted where we wanted it to. So we dye them, we study them. Those are the ovaries of Dalbolus that are filled with eggs. This is a control. These are the females that have been interfered. The egg inside the ovary, and then we studied that ovocyte, if there was a mismanagement in the cell, etc. If, in addition, we measure the level of transcripts at qPCR, we will also see that, the black column is control, the number of transcripts from day 1 to day 7. This is for injection, and the second graph for food, for feeding. It goes down, but not as efficiently as in the case of the treatment by injection. So how can we improve that? To think about this as a way to control the Dalbolus mighty. So we started the researching, and we saw that there was a lot of literature that spoke of ribonucleases or dsRNases, that degrade this RNA double chain. Of course, we went to the transcripts, we characterized, we seek for them. There could be just one, only one. There were 3. What should we do? Which is the gene to be silenced? Then, what we did was to see in which tissues they appeared. So, and we say that dsRNase 1 had a fatty body in the same end, dsRNase 2. But in the number 2 is
El Dalbolus tiene un aparato bucal.
the Dalbolus has is very complex. It's got saliva, and the other allows them to feed, and starting degrading the RNA. So we silenced the dsRNase 2. So we injected the dsRNase, and then we feed our target gene, the one we were using, and it went down. It was wonderful. It worked wonders. So you can see there that only the one in the middle is with dsRNase 2, and we could improve it. But we wanted to be by feeding, so we did echo feeding. And when we saw this, we could see our control females, the number of eggs, the females with bicaudal, fed by bicaudal, and those fed on dsRNase and BRC. So we have a lesser amount of eggs that is similar to the ones obtained by feeding. And what we saw was not only that there was a lesser amount of eggs, but that many of them did not have an embryo inside. The same, we finished corroborating their efficiency, RNAi efficiency after co-feeding. We are CONISAT researchers. We have to publish a paper. We presented 2 papers, but in general, this is what can be said. The RNA interference technique by artificial feeding is functional in Dalbolus mitis and constitutes a potential method to reduce the population of this pest by interfering with fertility. This and several fertility genes are part of Lucia's thesis, but in the meantime, we couldn't study only the fertility area. There is another thesis that is focusing on immunity genes. We have more transcripts there that are quantitative at the different times of the latency of the valvulus. Another thesis is focusing on the different pathways. And another thesis is focusing on the pathways related to the change. To the molt. Let me show you a last picture. They are silenced with ETH, a hormone that it's part of the process, and from the adult they do not molt. They try to molt, but they get trapped in their own shell and end up dying. And we have a high mortality rate. This is regarding the interferent. On the other hand, we looked for resistant genotypes. This started in 2014, and then we published it in 2020. We tried to characterize hybrids with resistance to corn stunt disease. As you can see in this graph, with this hybrid, we're going to speak of a dark spot. We're not going to delve into what type of hybrid it is. I think none of these are in the market anymore. The dark circle hybrid is 6 hours. 6 hours later, had even more leafhoppers that went into that hybrid compared to the others. And as we pushed away, we see that there are more leafhoppers and the others lower their amount. That is to say, you must have heard about this. It's not the same any type of corn for this type of leafhopper. It's hyper-selective. In the second graph, we can see the same. The hybrid that attracted fewer leafhoppers, but if we see the incidence, and if we force them to get into that hybrid, they died much sooner. So they don't like it. If we let them wild and they are going to go to a neighboring field to see if they find the right corn, or otherwise they might die. But once we did all that characterization, we wanted to go one step beyond. We knew that there was a technique called electric penetration graph that allows us to understand the feeding dynamic of insects. This generates an electric circuit between the insect, the plant, and the computer that starts generating these waves. I'm sure, and some of you did an electrocardiogram, and you see the different waves. Well, this is fine. Well, we can see the same. Thanks to the width of those waves, we can interpret where the doublus is feeding on. Cinema, phloema, all of those. Here we have a first salivation is B, and C is phloema. what we do, luckily we could purchase equipment thanks to subsidy we received. After certain complications, we could finally import it, and the pandemic struck. I don't know if you can see this. This is a drop of glue that is silver-based, that is inserted on the back of the dalbolus, that is connected to a gold thread, and then is connected to an electrode that is up there, and another electrode goes to the root of the plant. There we generate the electric circuit, and we can see the waves on the computer.
So we took those 4 hybrids from the first work, and we retested them here. Of all these curves, once we started analyzing them, we could have more than 20 variables that we could measure with all of that. And I took a few, for me, the clearest examples. If you take a look, this is the hybrid that is preferable or preferred or susceptible. It's the number of probes per insect. Since they insert the probe until they take them out. So they had fewer number of probes, but they were longer. So this is the period for each probe. So what did that insect do in the hybrid that he liked or it liked? They inserted the probe, they took the xylema, they salivated, and they got to phloema, and since it liked it, it stayed in the phloema for hours. And when I measured it, it indicated much longer than the others. So they did a lot of probing, but they were short in this case. Why? The insect is forced to eat from that one. It's hungry and it's tied like a dog with a leash. Yes, so they have to eat from that. But what did they do? What did they do? They insert the probes, but they can't reach phloema. And if they don't like it, they take the probe out. So it's a lot of probing, but short ones. And so it shows me which hybrid is resistant and which one is susceptible. If you want to see all the data, you can go to the paper. So with this, we can see the difference between susceptible and resistant hybrids, but you can also see the effects of insecticides, of seed treatment, and also the preference for other hosts. And Dalbolus only feeds on corn, people say. But in this crop season, we had a lot of consultations. There was chaos. What will happen with winter crops? What happens with oats? I have a lot of Dalbolus on oats. I see that all the Dalbolus are eating the oats. We had the pots to feed the Kusheli. They were starting their thesis, and I said, grab the Dalbolus, grab the oats, and just sit in front of the EPG. And this is, of course, preliminary, and it was done in this period of time. We had about 20 repetitions. Each repetition is an insect eating for 20 or 24 hours, forced to eat those oats. And this is the extract from 1 hour. And this is in xylem. So we didn't find any salivation, and we didn't detect them eating from phlegm. However, they were disruptive, so it was very difficult for them to insert their probes. But that's what the word says. It's preliminary, so it's the first trials and tests, but the results are not in yet. With the healthy insects, I mean, we have other studies, but I will show them to you next Aapresid Congress. And finally, in closing, everything that we do in the lab, I have the two colonies, inoculative, healthy, and so as a routine we do PCR to detect whether they are healthy and that the plants that are going to feed the colony, so that we have the real inoculatives really have the spiroplasm. So the control was very well implemented. So we sat down with the people from CONICET and we put together a stand to offer the service of spiroplasma detection for the insects and the plants. This year we received thousands of samples of insects and plants. We started working with them, and I ended up being part of the network for the testing. I hope you weren't bored too much with this presentation. This is our humble contribution to tackle this chaos or this drama of Dalbolus milis. Thank you.
Bueno, muchísimas gracias. La verdad que es sorprendente todo lo que hemos...
Very surprising, Ines. Thank you so much. I was quite surprised. We're getting a lot of questions, but I do invite you to scan the QR code and continue sending them so that I have a lot of questions to ask the specialists. We spoke about the integrated management, the tip of the pyramid, but I already have 5 questions that have to do with chemical control. Thresholds, what is a threshold considered manageable to make a decision on chemical control? And how many leaf hoppers per plant is considered a controllable population with agrochemicals? That's the million-dollar question. How many leaf hoppers do I have to apply that? And that is really a question that usually does not obtain a concrete answer. I mean, this is a vector, and the things change throughout time. So it is actually a question without one solid single response, as we could have with other pests. You know, you have to wait until you have 5 or 10 leaf hoppers. It's very important to reach high populations. I mean, in the last crop season, with 2 leaf hoppers, it was already very concerning. And this can take a toll on the crops. So basically, I think that that's where the most important support is needed, to do the follow-up of the crop. We know that this is a crop that evolves when it has the proper conditions, and it evolves quickly. So maybe from one week to the next, you can find a completely different scenario. So we would have to reinforce much more when you go to the field and you check and you verify. I mean, we can think like with other pests. You cannot say, well, let's see, I'm going to go get the product and see who applies it. You have to have a strategy already designed so that when there is a need to react, you react as quickly as possible. Another question, more details about crop monitoring. Which traps? Is it only with traps, and from what stage? Traps are just one more tool that will contribute to the monitoring of this pest. So we need to continue looking at these crops. We won't just, be guided by what the trap says. It can be a help, it can be a complement, yes, but the basis, as with everything and with all management practices, we have to be on top of matters. So the trap can be a support, but we have to be more aware and go out there to the field and detect at an early stage the occurrence of the vector. Question about the data that you showed. Why that reduction of efficiency from April to September? Why the reduction of efficiency if there's not a new generation? Many have asked that question. Actually, when we started working with Mine, we started in September last year. So it's not September of this year yet to say, yes, in September it goes down, or that factor makes it go down in September. The data that we have are April and May, and we have that data in that area. But, we have to see if that is repeated. I don't know if this is always the case. We still have data missing. We still have to analyze a lot of samples, and I think that is a little bit the work that we have ahead as a network. We still don't know what happens after September, so the very high levels that were shown, and when we saw the level of infectiousness, I called Alejandro and Augusto because we were worried. The PCR doesn't fail, and if you have the controls, we have reliable data. But these were leafhoppers that had been collected at a time when all of the corn and all of the lots had spiroplasma. So most of the leafhoppers were probably eating that corn and will probably be carriers of spiro. So what are we going to find in the next samples? We will tell you the next time. We have to continue collecting information. The infected individuals, do you do a molecular test in the field? No. Infected? No. That's why we do PCR. Do you want to discuss how the system will work? In the national network, we've initially discussed monitoring of 450 localities in all of the corn areas of Argentina, and 10% of those localities had to be representative of the region. And so what we did was to collect adults of Dalbolus mites seasonally. So we have 4 collection moments in the year. They are sent to Ines, and that's where she analyzes the samples. And Ines, for you, there are several ones who are similar. Can we extrapolate that technique to the field? They're talking about silencing. There are several ways. Initially, some years ago, what we had thought was to modify the corn plant so that the plant could express this double-chain RNA. There's one for Dalbolus, but there is a technology that works that way. And we had conceived it like that for Dalbolus. That is an option. And it's a modified plant to express double-chain RNA. We have Dalbolus, they eat and they get silenced. The other technique is spraying of this double-chain RNA, but that RNA is very easily degradable. So if we come here and we just spray RNA, 10 minutes in the sun and it is no longer useful. So we must protect it. encapsulated through a nanoparticle. And these are things where we are still working and that we're still working on. So I will get back with more information once we are more advanced. And the people at the university will, they put a legal muzzle on me, so I can't. Ines, you showed preliminary information on wheat. They want to know if it's true that they feed on brachiaria. And I didn't try it on brachiaria. On oats, we saw that it didn't reproduce. Days and days, and they die more quickly. But we weren't able to reproduce it. I haven't tried it on brachiaria. Maybe something to say about that is that some time ago we went to Brazil for this issue as well, and in some cases they mentioned that apparently brachiaria wasn't multiplying, but it just allowed them to live a little longer. With this issue of Dalbolus, we have things that they are feeding on something or other things. And in Brazil, they were mentioning that. Well, the importance of information. There are many species that do feed on that. The Dalbolus is there. But what will probably happen is what I showed you with oats. They drink water to avoid dehydration, and they can last up to 90 days. But they would use these plants to withstand those days. We have a red sign in front of us, but I want to ask a last question. What projections could we do depending on the population level of the current and the planting of corn in the north of Argentina? Well, we saw what happened in the last crop season with spring crops for corn, and this situation in the area, it's a practice that takes place in certain areas for spring crops. But in many areas of the country in the last crop season, we had a gap that was very broad. They started planting in September and they ended up ended in December. So the not knowing about that practice has made very many populations grow. So I think that what we should do is make certain adjustments in establishing that type of strategy in order to avoid this from happening, what happened last year. Maybe last year, and if we think of the same case of voluntary corn, who was thinking of doing control of voluntary corn? At the time, no one. But now, we know that we need to incorporate it. Okay, we have many questions left, but we will continue in the next presentation, so you can stay, and we can continue to see the available information for this problem. Thank you all. Thank you, Augusto and Ines.